online seminars. These are hosted by the Biostatistics and Health Informatics Department and the Prediction Modeling Group here at King's College. We have been celebrating these seminars for many years now. I have really lost the number of years. I think it's more than 10. And we are very happy to start this new season after the summer break. So these seminars will be happening every year the last Wednesday of the month. And if you have any research related with machine learning, healthcare, prediction modeling that you would like to share, I think that this can be your space as well. Not only as an attendant, but also as a speaker. We really, really welcome your presentations and that you share your research here. You can get very nice feedback from different people working on machine learning, on AI, and particularly with applications on healthcare. So you are really, really welcome. If you don't know me, I'm Raquel Iniesta. I'm a reader in statistical learning at the BHI department, and I'm hosting these meetings together with Professor Daniel Stoll. And today we are very happy to start our season with this is an exceptional date. This is actually the first Wednesday of the month instead of the last. And we are inviting Oli Payne, who is already with us. Oliver, thanks very much for accepting our invitation. And we are really looking forward to hearing you. You have been, as we were saying before, really popular recently with this the work of the publication of GenoPred. And this is the work we're going to be sharing here with us today. Exactly the title of your talk is Polygenic Scores in Practice, Standardization, Missing Variants, and Lessons from GenoPred. And as a short biography of our speaker, Oliver Payne is a Sir Henry Wellcome postdoctoral fellow at King's College London. And his research focuses on the development and application of statistical genetics methods to improve the accuracy, equity, and clinical utility of polygenic scores. He has led the development of GenoPred, a reproducible pipeline for polygenic scoring, and he's collaborating widely across international consortia on genetic prediction of complex traits and treatment response. So just let me tell the audience that we're going to be recording the meeting. Liz, thank you for starting the recording. I hope none of you have a problem with this. If you do, please just tell us on the chat. But in principle, this will be recorded so that some people that couldn't attend can see it later. And if you have any questions at the end of the talk, there will be a dedicated space for questions and answers. So let's start when you want. I hand it to you, Oliver. Thank you.
Thank you very much, Raquel. Thank you for the introduction and the invitation to present. So today I'm going to be talking about polygenic scores. I'll introduce what they are in a moment, but then how can we use these kind of biomarkers in practice? So first of all, I'm just going to introduce polygenic scores, talk a bit about the GenoPred pipeline, which is a software pipeline I've been developing for polygenic scoring, and then a new project that I'm working on right now, which is kind of looking at the next step of how do you then share your polygenic score models and apply them in external data sets in a kind of consistent way. And I think this applies well to other areas of prediction modeling as well. So a little bit about polygenic scoring for those that haven't heard of this before. So hopefully, as we're aware, is genetic variation explains a large proportion of individual differences across a range of health-related outcomes. For example, in our risk of disease or, let's say, response to medication. And we've conducted, as a field, hundreds or thousands even of genome-wide association studies, these GWAS, which look at common genetic variants across the genome and test whether there is an association with our outcome of interest or phenotype. And this, you basically run millions of regressions between each SNP and the phenotype of interest, and you end up with what are referred to as GWAS summary statistics, which show the association between each genetic variant across the genome and your outcome of interest. So, for example, each row is a genetic variant. There's some information about where that variant is, what its ID is, which allele at each location you're looking at, and then an effect size, some error, sample size, p-value, etc. So this is what you get out of a GWAS. And there have been many GWAS over the last 15, 20 years. And this is just a little animation just to kind of illustrate the point that over this time, there's been thousands of robust genetic associations identified across the genome with a broad range of kind of health-related outcomes. And this plot doesn't even go to today. I don't think it'd be possible to plot it. It's already kind of incomprehensible. But you know, all of this data is publicly available. So we have these GWAS that have included tens of thousands, if not millions, of individuals in them, and the results of those studies are publicly available online. So an amazing resource. What do we do with them? Well, one of the things we can do is polygenic scoring. So this is where we take, so a polygenic score summarizes an individual's genetic risk or propensity for a given outcome. And in their simplest form, they're calculated as the GWAS effect size weighted sum of risk variance. So you look at an individual, look at what genetic variants they carry, and then see whether any of those variants have an effect on your phenotype of interest, and you kind of add up all those effects, and that's your polygenic score. And these polygenic scores have a range of uses in both research, but also personalized medicine. So I'm going to give some examples of its uses, particularly in personalized medicine, because I think that's probably more of interest here. We could look at prevention. If we can predict the risk of disease in advance of it occurring, which we can with polygenic scores because they are stable across the lifespan, we could potentially intervene earlier and prevent disease if we know how to. Alternatively, when a patient comes to the doctor and they get a diagnosis, you could use their genetic information and these polygenic scores to help you refine the prognosis to say what's the course of that diagnosis going to be like. And also you could use this information to guide the treatment plan for that individual. Say, avoid this drug in case you'll get these side effects, or maybe this drug will actually be more efficacious for you. And there's examples of polygenic scores already being kind of shown to be useful for various of these applications. This is one study which is kind of, it's used quite a lot as a reference, but because it really nicely shows that these polygenic scores can actually stratify individuals in terms of their risk for a range of common diseases already. And they can do so to a kind of equivalent extent of these rare genetic variants that are already used in the clinical setting. So that, kind of begs the question, we should be using these polygenic scores, and we could be using these polygenic scores already. So just to illustrate this, on the figure on the right looks at coronary artery disease, and it's saying that individuals in the top 8% of the polygenic score distribution for coronary artery disease actually have a 3-fold increase in their risk of coronary artery disease compared to the general population. So that's quite a large proportion of individuals to be identified using a polygenic score at a substantially elevated risk. This has been carried forward. So this was a clinical trial where they looked at using polygenic scores in addition to a currently used risk calculator for QRisk2 to predict the risk of coronary artery disease or cardiovascular disease. And this showed that polygenic scores improved the prediction of risk significantly over the QRisk2 kind of risk calculator, which is important to establish. But also this additional information was kind of accepted by the clinical practitioners as well as the patients. So this information both improved the accuracy of our predictions whilst still being interpretable and kind of explainable. So apart from predicting risk, we could also predict other things, as I mentioned, for example, predicting response to medication. And this was a nice study where they took clinical trial data for the use of statins to treat coronary artery disease, again, and they show two things in this figure on the right. One is that individuals with a high genetic risk for coronary artery disease, first of all, had a higher overall disease event rate, such as heart attacks, during the clinical trial, which you would expect, but also the reduction in risk caused by being given statins compared to the placebo was substantially greater in those with high genetic risk. So essentially there's an interaction between the polygenic risk score and the effect of statins. And this, again, kind of helps us see a possible applications for polygenic scores in terms of identifying individuals that might respond to a given treatment. And this could also inform clinical trials and things like that. This is just another example that came out this year. I should say all of these studies that I've just mentioned are within the last 2 years. So this is really just emerging right now in the clinical kind of setting. So it's quite exciting. This one came out very recently this year, showing that polygenic scores again can improve detection of risk for prostate cancer in this case. And it's quite a striking result. They showed that if you look at individuals in the top 10% of polygenic risk scores for prostate cancer, of those individuals, they looked kind of deeper by doing biopsies and showed that of the people that had cancer in that group, 72% of those individuals that had cancer would not have been diagnosed with their intermediate or high risk classified cancer if they had not had the polygenic every year strain scores can be used currently to improve risk stratification over and above the currently used clinical tools. So hopefully I've kind of convinced you that polygenic scores already have clinical applications. So what are the barriers that are stopping this form of biomarker from being rolled out for everything in healthcare? Well, there's 3 kind of barriers that I think there's probably more. But one is that, of course, we're not able to accurately predict many interesting clinical outcomes. I've talked about risk of prostate cancer and coronary artery disease. We can predict risk of kind of, I would say, well enough for a handful of other traits, but there's many clinical outcomes that we can't yet predict. For example, likelihood of response to antidepressants. And there's many other examples of that. And this kind of poor prediction accuracy is particularly a limiting factor in populations of non-European ancestry. And this is because many of the genome-wide association studies have been conducted primarily in European ancestry participants, and the transferability of those findings is often imperfect. So we really need to identify novel methodology that can leverage the data we already have to the best of our ability, and of course we need to collect more data, conduct GWAS on more clinical outcomes of interest, and include ancestrally diverse participants. Another barrier is the interpretability of polygenic scores. So a polygenic score is influenced by an individual's ancestry, and that doesn't necessarily indicate that they have a different propensity for the outcome that you're trying to predict. It could just be biased by the fact that different genetic variants exist at different frequencies in different populations. So it's important that we standardize our polygenic scores according to an ancestry matched reference. And that's not always that easy. Another kind of part of this interpretability point is calibration. We need to be able to convert our polygenic score into a calibrated risk estimate to say, what does this mean in real terms? What is the individual's absolute risk given their polygenic score and other clinical variables? Finally, I think one of the main barriers is the expertise required to accurately calculate these polygenic scores. It requires a lot of very large data sets and complex data sets, and the methodology is advanced and also rapidly evolving, which makes it hard for someone who's, say, running a clinical trial to be able to easily calculate these polygenic scores in a reliable way. So this is where kind of GenoPred came in. So GenoPred is a research program I lead in collaboration with many other people at King's College London, in particular Catherine Lewis, and this project kind of focuses on several things. One is benchmarking existing methods for deriving these polygenic scores to figure out what works best in different scenarios. Another aim is to derive and evaluate novel methodology to try and kind of improve the accuracy of our predictions, particularly in those of non-European ancestry. Another aim is to kind of develop a pragmatic framework for developing interpretable polygenic scores, and finally to develop easy-to-use software implementing the best practices. And this is what we've kind of been doing over the last few years, and I thought I would summarize 4 studies that have helped achieve these aims and have kind of relevance to wider interests, hopefully, of the prediction modeling kind of audience. So this was the first study we did looking at polygenic scoring methods, and really it was to find out which method works best across a range of clinical phenotypes. And this helped kind of guide researchers in future studies to select the method that would work best for them. But one kind of nice finding from it was that actually we used the trusty elastic net, and we showed that if you fit polygenic scores from multiple methods in an elastic net model, it actually maximizes prediction across all of the traits we looked at. So this was kind of a useful study for guiding future research. They're now referred to as ensemble polygenic scores. These models that combine polygenic scores from multiple methods, they are now kind of, I would say, it's still, it's up for debate, but they typically perform better than any single method. And we also documented all the code and made it well publicly available to help kind of other people start using it. Another study that we conducted was addressing this issue of poor performance of polygenic scores in non-European populations. So what the study was doing was looking at how we can best leverage the data we have to date. So we have very large European GWAS, and we also have now emerging GWAS in non-European populations that are usually of smaller sample size. And this study kind of looked at how can we leverage both these types of data together to maximize prediction in the non-European populations. So we kind of benchmarked a range of methods to do this, identified which was working best, but also developed a novel extension which made it easier to use these methods, both in terms of the kind of computation time, but also we adapted it so you don't need individual level data to tune your polygenic scores, making them kind of more usable. So the third study was looking at this question of interpretability, where we kind of talk about this process of standardizing our polygenic scores according to an ancestry matched reference. So you can then look at your polygenic score in units of standard deviation from the mean, like a Z-score. And then from that, we developed an approach for converting those polygenic Z-scores into kind of absolute estimates of risk or absolute estimates of a continuous phenotype. So we did this for both binary outcomes and normally distributed outcomes. And all you need to give it is the variance explained by the polygenic score and then the expected distribution of the phenotype in your target population. So the prevalence, say, of a binary outcome. And from that, it could tell you, okay, if you're in the, let's say, 98th percentile of polygenic score risk for schizophrenia, that changes your absolute risk from 1% to maybe 2%. So it helps put things into perspective, and that's, you know, incredibly important if you're going to actually act on this information. And then this fourth study, kind of illustrating GenoPred, was where we published a software pipeline that provides a kind of easy to use snake based pipeline implementing the leading methodology as based on our research and other research at the time, and it's ongoing, still developing it. That's been tested extensively, it's computationally scalable, and it's also reference standardized. And I think the prediction modeling kind of audience here will appreciate this a lot. The way it works is reference standardized, meaning the polygenic scores that it calculates are not derived in a way that is specific to a given target population. They are standardized according to a common reference data set, and then polygenic scores can be calculated in a very consistent way in any target population, and that makes the kind of interpretability, well, it makes the polygenic scores more kind of comparable across different scenarios, helping improve both the kind of reliability of findings, but also the interpretability of the score. You know, one angle we put a priority on was making sure this software could cope with data from populations around the world. This has helped a lot of people, I think, use polygenic scores in their research, which is amazing. And I'm still using it with various collaborators to continue answering various research questions relating to polygenic scores. So we've overcome many barriers. This is great progress. There's still a lot more to do. And one issue that I haven't looked at yet, but is something that's become kind of clear more recently, is how do we, once we have our polygenic score models, so the SNP weights that we use to predict, to calculate these polygenic scores, how do we share them and how do we apply them in external data sets? So try and give a little bit more kind of clarity on this. So a polygenic score file is kind of like your GWAS summary statistics, except the effect sizes have been adjusted in some way to make them better for predicting a trait of interest. And there's, you know, a whole load of methodology that could have gone into creating this score file. But essentially, once you've got your score file, you've done your study and you say this score file can predict coronary artery disease very, very well. What do you do then? Well, you can put it online, right? So there's this repository called the Polygenic Score Catalog. There's other databases you could upload these score files to. And that's brilliant. It means you can use these score files in theory for future studies. But actually, there are a number of issues that can arise at this point of trying to use some previously published polygenic score model. And I think, you know, just think of this in terms of these issues, different technical issues, but similar kind of impact would occur when, say, some study publishes their amazing prediction model and puts their model online, and you know, sometimes they don't perform as well as you hoped for various reasons. And this is the same issue, but they're kind of specific to polygenic scores, and we might be able to resolve some of them. So what are these issues? What could go wrong? Well, so one of the first things is, unfortunately, a lot of the score, well, some of the score files that people upload don't have sufficient information to actually reliably determine what genetic variant the effect size corresponds to. And these are referred to as unmappable, meaning they're variants that we can't actually identify in any common reference database. And that means they're effectively unusable. Another issue is strand ambiguity. So I'm not going to go into this too much because it's a little bit technical, but essentially, DNA is has two strands, and we are always looking at one or the other strand, the forward or reverse strand. And when a SNP or a genetic variant has what are called ambiguous alleles, it means it's not possible to see which strand you're looking at. And this is the case if the alleles are A to T or C to G. And essentially, this also brings up a problem because we actually don't know which strand we're looking at. We don't know whether this effect size corresponds to the A allele or the T allele. And, there's ways of resolving that, but it's a source of potential kind of mistakes. Another issue is that people often don't document what the other allele is. So we know that the effect usually corresponds to the allele in the A1 column, but if they don't tell us what the A2 column is, we don't know what we're comparing against. And we can sometimes infer this information by saying, well, if it's not a G, then it'll be a T. But sometimes in one site in the genome, you can have multiple alleles. So you don't know what the alternative allele is, and again, you have to drop this information if they don't tell you that. So these are all kind of sources of kind of ambiguity that mean that would actually just lead to these results being unusable, which is quite drastic, but it happens. Another issue is that when you then look in the target genetic data set, some of the genetic variants that are in your score file may not actually be present in your target genetic data set. And we need to know what to do in that scenario and how to minimize that issue. And a related issue is that if the variant was missing, you can sometimes impute that missing data, but the quality of that imputation will also impact the quality of the end result, the polygenic score. And essentially what this means is you've gone from a polygenic score that's been validated in some previous study, the score file's been put online, but then due to all these kind of small little things, you end up with a polygenic score in your target sample that represents something a little bit different. The main impact that I'm interested in is the reduced variance explained. So the predictive utility of this score is going to be degraded by all of these sources of ambiguity. So really the aim of this current study was to quantify, to see how often these issues arise in real data, quantify their impact on the variance explained by polygenic scores, and then think about how we can improve the applicability of polygenic score models that are published in the future. And I must say, this work is ongoing. These slides are not yet fully developed, but I think it already shows the issue quite nicely, and I'll do my best. So what I did is I downloaded coronary artery disease polygenic score files from the PGS catalog, of which there were 78. And just as a kind of descriptive, these polygenic score models can range massively in terms of how many variants they contain. Some polygenic score models consist of only 7 variants, and the most included in this list of polygenic scores was 7 million variants. So, the vast majority usually have fewer than, 1,000. variance, but I would say more and more going forwards in this kind of in using today's methodology for polygenic scoring, we're going to see a lot more with millions of genetic variants included. So what I did is I looked at how many variants do we lose due to this issue of them just being unmappable, no other allele information, or being strand ambiguous. And of the 78 polygenic scores that I looked at, 41 of them lost over 5% of the genetic variants they included just due to this basic filtering. And that's quite a lot. In some cases, over 40% of the variants in the score file couldn't be used, which is going to dramatically impact the variance explained by that score. Of course, there were many score files that were very well documented and there was almost no loss, and that's important. But we need to kind of figure out, how we can avoid these variants being lost in general. I've broken down why they were being removed in this figure on the right. And the largest category here is strand ambiguity, which is potentially avoidable, and I'll talk about that on the next slide. But the other kind of massive loss of data was this unknown A2 or other allele information, which is very avoidable. It just requires people to upload that information, and it's so that's a good lesson learnt. So this strand ambiguity thing, I won't go into it too much because I'm aware this isn't maybe a massive interest to a lot of people here, but essentially what I'm showing here is that a lot of these variants are strand ambiguous, which we could say means we can't use them. But actually, if you look at the non-ambiguous variants, the vast majority of them are on what's called the forward strand. And that's what we would expect. Most data is on the forward strand. So it's arguable we could use these ambiguous variants and just assume everything's on the forward strand. Is that a safe assumption? I think that's up for debate still. The many tools, in fact, as far as I'm aware, all tools by default will throw these variants away. So I think it's a good point to kind of look at what's going on now in the literature and the data online. Is everything consistently now on the forward strand? If it is, then maybe we can say kind of recover these variants. So another issue that I was looking at was this issue of having missing data in the target sample. And I've been looking at this in a couple data sets where I have information on what the imputation quality is and the availability is of variants across the genome. And essentially from that information, I can infer what the decrease in the variance explained by the polygenic score would be. I won't go into how this works, but it accounts for LD, which is the correlation between genetic variants, which is an important thing to note. And essentially what we see, there's a couple of things, I'll break this down. So with the remaining variants, so the ones that weren't lost due to kind of the bad metadata, I suppose you could kind of look at it as, of those, 96% of the variants are common and have a kind of decent level of imputation in the UK Biobank. And this is just a kind of, the reason I'm mentioning this is because this is where I estimated the LD, which I needed for the next step. But I think the fact that only 96% of them are actually common and well imputed is a bit of an alarm bell, because it means 4% of those variants just are likely to be poorly imputed, and that will degrade the quality of the polygenic score. The next thing is, of those variants, we can then look at how the imputation quality across different panels and different imputation panels and different arrays will impact the polygenic scores. And you can see in this figure down here that across different arrays and different imputation panels, the prediction accuracy or the relative accuracy is fairly high. It's always above 90% on average. But for some scores, it drops dramatically, and it's consistently imperfect. The average is always, even the highest average is around 98%. And that sounds very good, and it is good, but we should be making this 100%. There's really no reason it shouldn't be. And that's just broken down here in this histogram. The vast majority are doing well, but there's quite a few that aren't, and we need to figure out if we can make them more generalizable than they are currently. This was using some data from a kind of imputation metric database, but I also looked at some real world data that I've been using as a part of a project for the Psychiatric Genomics Consortium, and you get the same result essentially, that the transferability of these polygenic scores is consistently imperfect, and in some cases it's quite poor. So one of the reasons for this is the selection of variants that we're using in the scores. Historically, people have been using kind of bespoke lists of variants that are much harder to find in other data sets because they've maybe used an array that's specific to a disease, and that's fine. We can still use those, but it will impact the likelihood of you being able to use that score in a data set that hasn't used that bespoke array. But more recently, methods use one of two lists. Either this HapMap3 list, which is around 1.2 million variants, and it's used because it provides decent coverage. They're considered well imputed generally, but I think the main criticism I get when I still use it is, well, it's not very good coverage. The genome is much more kind of dense than that really gives it credit for. So more in the last year, in fact, this new method called SBase-RC has been published, which scales this up and now includes 7.1 million variants. And it defines this list because they are common and they have decent imputation in UK Biobank. And they show in their study that this improves the predictive utility of polygenic scores in UK Biobank and some other data sets. But I think something, and this was one of the inspirations of this project, was that how well are these variants imputed in other data sets? And although using this 7.1 million variant list is good in some settings, the poor imputation may actually undermine those kind of methodological advances, just purely because you're using variants that we just don't have in other data sets. And essentially, we find evidence of that. So this is looking at kind of imputation quality across different arrays and different imputation panels. And what we can see is that, yes, the average quality of imputation is lower when using this larger set of variants. And I think it's hard to see really in this because they're both so close to 1. But if you look at these bar charts on the right, you can see that, there's about maybe, I don't know, even with the best array and the best imputation panel, when using this SBase-RC list, you're losing maybe, you're imperfectly imputing over 10% of the variance in that SNP list. Whereas the HapMap3 list, it's less dense, but it does consistently have better imputation. So I think we need to kind of look at the relative gains in terms of coverage of the genome and generalizability to external data sets. So one thing left for me to do is potentially define a better list of variants for us to use by looking at which variants are well imputed across arrays and imputation panels, and ideally populations. This is an issue that hasn't been talked about in previous literature, but we need, if we can use variants that are well captured across populations, that will also mitigate the issue of using European data to predict in non-European populations, which is, if not addressed, going to lead to kind of greater health disparity. So some brief conclusions. Essentially, and these, essentially, we need to make sure the score files and the models that we're sharing are well documented, and that is easy to do. GenoPred, as an example, already outputs the data in a highly standardized and well documented format, which can then be uploaded to the PGS catalog. And you can use different sets of variants. You can use the more well imputed HapMap3 variants. Soon I will release a version using hopefully a denser set of variants that are also well imputed. And that will mean that the scores coming from GenoPred are going to generalize better to other data sets. And I'm not saying you have to use GenoPred, but it's an option. It's not that straightforward calculating these scores. And I think something really important is when you're calculating your polygenic scores in a target sample, if there is missing data, we need to quantify how much missing data there is and the impact that's going to have on the polygenic score variance explained. And that's what that method I presented does. It's a way of kind of estimating the relative performance of your polygenic score given the data you have in your target sample. And that's going to be important for kind of calibrating the predictions you get from that polygenic score in your target sample. And ultimately, if we can adjust our score files to use the variants that are present in our target sample rather than the ones that are missing, that would be great. And I'm aware of some methods that are being developed for that currently, but none are available to date. So I'll end it there. I would say a massive thank you to everyone that supported me and the development of GenoPred, including Catherine Lewis and her team, as well as those that sponsor my fellowship, mainly Ammar Al-Chalabi and his team, as well as sponsors that have supported me along the way, and the funders, of course, the Wellcome Trust and the NIHR BRC. Thank you.
Thank you very much, Oli, for this excellent talk. I was just wondering if there can be any comment or question from the audience. I think that Gareth Williams has raised his hand. Gareth, when you're able.
Hi, hi there. Thanks for the talk. It's very interesting. I mean, I'm not an expert in the field, but I have looked at some of the PGS stuff, and I mean, it appears that there's this kind of asymptotic behavior that no matter how many, if you increase the number of SNPs by a factor, you don't really increase the predictability. There seems to be this shortfall between for the SNP for the PGS-based approaches and actual known genetic susceptibility in a population. And I mean, lots of people have using a simple the PGS is just an additive weighted model. People have looked at, I guess, pattern recognitions, random forests and neural networks and things like that, and they're still not cracking this asymptotic behavior. So just wondering if you think the problem is not so much the covering and how we analyze the existing data, but the fact that we don't have enough actual human samples, or do you think it's some other problem? I mean, I'm just speaking about the disconnect between what we know is the case, which we know that for instance, height is it's got a certain amount of that, the variance in heights explained by the genetics, but the polygenic risk is falling far short of that. So I was wondering what your thoughts are on how we actually bridge that that gap, which doesn't seem to be bridgeable at the moment because things are asymptoting. Yeah.
Yeah, it's a really, really good question and a difficult question, but I'll do my best. I think you're right. This I'll start the question of the kind of density of the coverage of the genome that these models have. This study by S-Bayes RC about S-Bayes RC, they showed that actually you could improve the variance explained by polygenic scores by using a set of variants that provided denser coverage, but only if you also considered the functional annotations. So what are the functional kind of impacts of these variants? Say, are they in enhancer regions? Do they, kind of information about what the variants do? And they found that there was an interaction between the density of the coverage and the inclusion of these functional annotations, which I thought was really interesting. And that's one bit of progress. And they also found in that study that the SNP-based heritability, so the estimate of what would the maximum a polygenic score be able to explain, was also slightly increased when you allowed for these functional annotations. So I think that's one angle that's closing this gap between the SNP-based heritability and the twin-based heritability, which is kind of the very, very upper limit, possibly even beyond what's possible. In terms of additivity, I completely agree. All of these models assume additive SNP effects, and there must be non-additive effects. There's evidence saying there is, but I think it's really hard to model them because of the scale of the genome. But I think also the problem is the additive model captures non-additive effects often really well. So trying to prove that the non-additive model actually does better and that these non-additive effects exist is hard because the additive model does so well. But I think you're right. There's definitely going to be extra variance to be explained if we were able to model those effects properly. I think the main factor limiting the variance explained by polygenic scores for most traits is the sample size of the GWAS. That's not true for everything. Height, I think as you said, they've kind of reached the limit of what you can explain with a polygenic score right now. But for many traits, that's not true. And I think that's probably going to be, as well as the methodology, one way to improve the sample size is to collect more data, but another way is kind of aggregating information from different sources that we already have from different populations, or for different traits that are correlated. And I think these two angles of increasing GWAS sample size and kind of fancy methodology will hopefully make it, clinically, will provide clinically useful variance explained for more traits using polygenic scores.
Thanks. Great.
Thanks, Oli. We have a few more questions. I think that Liv would like, sorry if I'm mispronouncing your name. Can you just go ahead, please?
Hi, it's just Liv. It's Liv Klein.
Okay.
I have a couple of questions, but I'll start with the one just about polygenic risk scores rather than kind of going a bit too far. I think so. By generating polygenic risk scores, you're ultimately generating the risk of developing X. So, or not the risk of. So, like the potentiality of being X height or developing
Sure.
cancer or yes. So how do you even define that X, especially if diagnosis is a bit more ambiguous, like in mental health, like depression is diagnosed by self-report and only a few symptoms out of a larger list need to be present. So I was working with polygenic risk scores and depression, but I know that there is polygenic risk scores developed for MDD, and then there's also PGS developed for depressive symptoms. So how do you choose what to go for and what would be the most informative?
Yeah, great question. So I think there's different ways to look at this, but it's, if you're trying to predict a specific outcome, for example, prostate cancer, then using a GWAS of prostate cancer, cases and controls, lifetime diagnosis of prostate cancer would probably be the best starting point. But then for some phenotypes, that limits the sample size a lot. So in the case of depression, it's much easier to use kind of less specific definitions of depression than a clinical diagnosis of depression. So sometimes, we have the clinical depression diagnosis GWAS. That's great, it's really specific, but it's limited in sample size, so we can scale up massively if we just ask everyone in UK Biobank, have you ever had depression? And you get many more people. It's not as kind of clean, it's not this clinical cohort, but it can actually provide better power when deriving your polygenic score. And I think ultimately, for your question, is you need to try different polygenic scores out. So if you have a data set where you have the phenotype you want to predict, or you have a GWAS that's specific to the trait you want to predict, then looking at how well polygenic scores from a range of traits, both for your trait of interest, but also secondary traits that may be genetically correlated to your trait of interest, see how those correlate in your target sample. So you could do that with polygenic scores and just say, how well do these polygenic scores for all these different traits predict my outcome of interest? Or you could look at the genetic correlations using GWAS summary statistics between your trait of interest and the other GWAS that could be correlated. So yeah, that's the way I'd go about it.
Right. Thank you. Can I do my quick second question? Because it kind of follows up on that, because you were saying, so looking at a broader population, so looking not necessarily as the diagnosis, and then you're getting a greater power because you're looking at a bigger population, but then environment is highly correlated, and that's essentially what is then gonna interact with that. So my second kind of question is that if the risk score gives the prediction, the chance, it's still not that 100%, especially if you're looking at a broader population and only symptomology. What would be the steps to identify then that environmental trigger, and then translate that for prevention, essentially? Or is that going too far?
No, I think that's really interesting, and I think it's not easy to answer. I think we need more research on this, to be honest. Currently still, we're usually thinking of these things as additive, and it'll be, okay, you have this much genetic risk. This means if we now throw in a bit of environmental risk, you'll be pushed over this kind of liability threshold. And that does seem to fit in most instances, this kind of additive model, but I'm sure there are going to be instances where there's some sort of interaction to say you're at particularly high genetic risk if you also incur this kind of environmental risk factor. But yeah, as of yet, I'm not sure there's any kind of robust examples of that. But yeah, there's different models of trying to kind of combine these different sources of information. Usually they are additive. But yeah, more work to be done, definitely. So yeah, I hope more people start including polygenic scores in their prediction models, basically.
Yeah, agreed. Thank you so much.
Thank you, Liz. We have a couple of more questions in the chat. The first one is from Diana. So she's saying, thank you so much for the talk, very informative, and the algorithm is very useful. My question is, what is your opinion on the final output of a polygenic risk score model and their unification or calculating population-wide percentile that could then be used in smaller studies? Right now, we've used GenoPred data to obtain some product of log odds, while other methods give neural network scores, and these outputs themselves may be on a different scale or differently calibrated to the probability of a trait. Long question. You have it in the chat if it's easier for you.
Yeah, I was just having a look. Thank you for reading it.
But that's not an easy one.
This is a challenging question. I think I generally try not to assume too much about the scale that the SNP weights are provided on. So some polygenic scoring methods output SNP weights that are well calibrated, allegedly. So they're on the polygenic scores should be on the same scale as the phenotype they're trying to predict. But not all polygenic scoring methods have that that kind of well calibrated. Like I think, to be honest, we need to figure out which methods do provide well calibrated polygenic scores. It's been suggested to be true for several of them, but I've not actually seen much demonstration of that. I think the best approach right now is when you get your polygenic score. So regardless of the weights, once you've used those weights to calculate the score in your target sample, then scale it according to your ancestry matched reference to get it in units of standard deviation from the ancestry matched mean. And then when you want to calibrate, I think either do that using individual level data where you have the observed phenotype, so you can figure out what the relationship is between the score and the outcome, or you can use the summary statistic based approach that I mentioned, where you kind of take into account the variance explained by the score and the distribution of the trait you're trying to predict, and then you can kind of infer the meaning of the score in absolute terms. So yeah, that's where I go about it currently. But I think it's an important kind of point is which of these methods provide calibrated scores. And I don't think I avoid it and assume none of them do and just go about it a different way. But I think some of them can, and we should probably be kind of highlighting those.
Thank you, Oli. We have a final question, still 1 minute to go. I think we can just cover that one if we go the final one. And it's a question by Guillermo Eduardo Parada Gonzalez. Thanks, Guillermo. Great talk, very interesting. And the question, do you see any hope for incorporating the effects of rare and ultra rare variants into polygenic scores?
Yeah, so there's I think polygenic scores and the methods that are used to derive them really do rely on the variants being common. But I think so the most kind of immediate option for integrating them is to just model them separately, because these rarer variants usually have larger effects. So usually people just fit the polygenic score and then have separate variables indicating the status for those rare variants. It's not a very satisfactory answer, but I think also rare variants have very little LD, and they should be fairly uncorrelated with the polygenic score. So I don't know if modeling them together using summary statistics really makes sense anyway. So yeah, I would say yes, you can absolutely integrate them together. It just needs to be done kind of using some individual level data rather than, say, from GWAS.
Okay, thank you so much, Oli. I think that's the last question. It's 4 now, so let's finish the seminar here. Thank you very much. That was absolutely excellent, and wish you lots of luck in any other future work that you do, Oli. And thanks all the attendants for having been here. This week, this month, sorry, we will have another meeting by the last Wednesday, so that will be 29th of October. And maybe some of you will know that sometimes we interact with the PPI group here at KCL as well, at the BHI department. And so the next talk will be in the intersection between prediction modeling and PPI. So if it's of your interest, you can join us on the 29th from 3 to 4 online again. Thanks, everyone, and enjoy your evening today. Thank you.
Thank you very much. Thank you for the great questions.